Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome Karine Le Roch from University of California at Riverside on this show. Please let me
briefly introduce you to our audience. You got your PhD from the University Paris-Sorbonne in 2001. You then moved on to do a postdoc at the Scripps Research Institute in La Jolla, California. You then became assistant professor at the University of California at Riverside in 2006 and associate professor there in 2011. Since 2015, you are professor with tenure in the Department of Molecular Cell and Systems Biology at the University of California at Riverside, and since 2016, director of the Center for Infectious Disease and Vector Research.
A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
It was a long road to science, I will say. I was an undergrad at the Sorbonne University. I was supposed to actually my undergrad was in biology, biochemistry, and chemistry, and I was supposed to do a master of biochemistry, and I was not that excited about it, so I decided to take a break and to do an internship in a lab in North Wales, Bangor, because I figured out that if I really wanted to stay in this field, I had to learn how to speak English, and I had to be a bit more excited about science. At that time, I was not that excited. So I ended up, by chance, in a lab working on a parasite, Schistosoma mansonii. It's a parasite. It's a little worm that can infect humans, and through the a complex cell cycle between a snail and human. And I became fascinated by this parasite and fascinated by the regulation of their life cycle, and decided to stay in that field. And since then, have not changed my field. I've been working in different parasites since the mid-90s, and I'm really excited about it. So understanding the life cycle of this very complex parasitic organism was something that I fell in love with.
Mm. So let's come to your science that centers around malaria chromatin structure and its transcriptional regulation. I want to start in the year 2003, and with the science publication from this year. There you used a high-density oligonucleotide array, I guess nowadays one would do sequencing, to generate expression profiles from human and mosquito stages of the malaria parasite's life cycle. So you kind of shared how you got into malaria, but maybe there's more to it. So how did you get into malaria research, and what did you find then in this paper?
So yeah, I started with Schistosoma mansonii and did my master after that in Pneumocystis carinea, which is a fungus infecting lungs of immunosuppressed patients. But it was not that easy to work with pneumocystis, and at that time there were 3 therapy for HIV patients coming in, and the funding was coming down for pneumocystis. So I decided to actually work on a parasite that was affecting a large amount of people worldwide, and that could eventually keep the funding for a much longer period of time. So I started in the human malaria parasite, Plasmodium falciparum. I did my PhD there on this subject, and again fascinated by the life cycle of this parasite. I move on to do a postdoc in the US with Elizabeth Winsler, where we developed this first microarray experiment covering the full genome of the malaria parasite. And because I was interested in the life cycle and life cycle progression, we decided to try to understand which gene were expressed at different stage of the parasite life cycle, and published this very interesting work that's over 20 2 years now.
Was there something that stood out from that? So were there some genes? So you looked at the genes of the malaria parasite, right? So not the host. Yeah.
We looked at the gene of every single gene encoded in the human malaria parasite across the full cell cycle, the full erythrocytic cell cycle, including gametocytes, sexual differentiation, early and late gametocyte as well. So the stage that are responsible for the transmission of the parasite from the human to the mosquitoes. And we also looked at sporozoite stage of the parasite that's responsible for the transmission between the mosquitoes and human. So the full life cycle, we couldn't really do the stage in the mosquitoes because they were very difficult to work with, and they are still very difficult to work with. Now it's a bit easier with next-gen sequencing technology, but at that time it was a bit more complex. So and understand and trying to understand. So once we generate all of these gene expression data sets, I become fascinated by trying to understand how this gene could be regulated, considering the fact that the human malaria parasite has a very small number of predicted transcription factor. And usually in higher eukaryotes, approximately 6% of the genome is responsible for coding for this transcription factor. In malaria parasite, it's absolutely not the case. It's not 6%. It's more close to 1 to 2%. So how the human malaria parasite was able to regulate all of these genes without a large number of transcription factor was something that I was interested by. And that's how I started to look at the role of epigenetic and chromatin structure in regulating genes in the malaria parasite.
Yeah, what you then did, at least this is my next question, what I looked up is that you followed this up by investigating the role of post-transcriptional controls in the regulation of protein expression. So is that what you just described, or is that a little bit different?
No, I'm always interested, and I'm still doing that. Always interested in understanding gene regulation and gene expression at the transcriptional initiation level. That's where I actually looked at now chromatin structure and epigenetic, and then I also looked at regulation of gene expression at the post-transcriptional level. So looking at the role of mRNA binding protein, regulating this transcript, or different type of protein modification, enzymatic change of protein, phosphorylation, ubiquitination, acetylation. So looking at gene regulation as a whole, and not only looking at transcription factor, but trying to kind of integrate gene regulation at the genome-wide level and looking at every stage of gene regulation from the initiation level, epigenetic, to protein translation, and also post-translational modification of histone or other type of protein.
Then, and I guess this was then from your own lab, so the first work from your own lab, you focused on the nucleosome landscape and how this controls the transcription of plasmodium. Can you maybe talk about the transition from your to your own lab, and then what you found in the study?
So that's actually my first work on the role of epigenetic and chromatin structure, trying to understand if change in nucleosome positioning could actually regulate gene expression, again with this idea that the plasmodium genome has a very limited number of transcription factors. So we first initially looked at nucleosome positioning, and I thought that I should be able to do this work using the microarray platform that we developed when I was a postdoc. But at the exact same time, the next generation sequencing technology was starting to emerge, and I decided to do this type of experiment using next-gen sequencing and the Illumina sequencing platform, trying to purify nucleosome bound, strongly nucleosome bound region of the plasmodium or nucleosome-free region, again at different stage of the parasite cell cycle and life cycle, to see if there was any significant change in this nucleosome positioning that could correlate to gene expression. And we did find significant change in chromatin structure, in nucleosome binding, that correlate with massive change in gene expression.
So this would indicate that there is chromatin remodeling going on, right?
Yes, of course. So we started by nucleosome positioning, and eventually later on chromatin structure change and epigenetic, looking at the change of histone modification that also modified the interaction of the nucleosome with the DNA.
Do they bring their own remodelers, or do they use like host remodelers?
No, they use their own remodeler. So the human malaria parasite is an eukaryotic cell, so he has all the machinery to deal with epigenetic and chromatin structure remodeling.
What kind of assay was that you did? Because nowadays one would do probably ATAC-seq, right? Is that comparable, or would you like to?
At that time, there was no ATAC-seq, so that's why we looked at nucleosome positioning. And now we can do ATAC-seq. There's several labs in the field that have generated ATAC-seq data to confirm that there is a significant chromatin remodeling to regulate gene expression. But I also do a lot of chromatin conformation structure. So I will say IC experiments to actually look at the achromatin and heterochromatin domain and do the 3D structure as a whole. So model the 3D structure. So the chromatin structure is a combination of I would say nucleosome positioning and histone modification, but you're going to try to look at the chromatin as a whole, trying to figure out which region are heterochromatin, so repressed region, or which region are achromatin open region, ready to actually transcribe gene. So we've been doing that again at different stage of the parasite life cycle. I keep the same theme in my research. We first use microarray, and then we move on to the next gen sequencing with nucleosome positioning and epigenetic, and eventually chromatin conformation capture with IC.
Yeah, we will come to the IC experiments in a bit. But first, you compared steady-state mRNA and polysome-associated mRNA levels of the plasmodium at different time points during its asexual cell cycle. So again, going back to the transcriptional regulation, so what did you learn from this about transcriptional translational control and gene expression?
So always with this idea of looking at gene regulation as a whole, not only at the initiation level, but also trying to figure out what's happening to this transcript when they are transcribed. We wanted to get an idea of what was the percentage of genes that were expressed or regulated at the initiation level, and which one will be the one that are expressed early but stabilized by mRNA binding protein. Because if the human malaria parasite like a lot of transcription factor, he has a strong enrichment of mRNA binding protein. So the idea was to try to understand what was the percentage of direct transcriptional regulation at this initiation level, and what transcript will actually sit in the cytosol and wait to being translated later on. So we did this comparison between total RNA sequencing, RNA-seq, and polysome-enriched RNA to figure out which type of RNA is associated with the polysome to be translated, again, at different stage of the parasite life cycle. And we find that approximately 50-60% of the gene are regulated at the transcriptional initiation level. So their transcripts are transcribed and they are quickly translated. But for approximately 40% of the transcript, they are transcribed at the initiation level, but they are sitting in the cytosol for a few hours, 4, 6, sometimes 12 and plus hours to be translated. So they are most likely regulated by mRNA binding protein that stabilize this transcript and release this transcript to the polysome fraction when needed, a bit later on during the cell cycle progression. So again, that type of experiments led us to conclude that there is a significant change in chromatin structure and gene regulation at the initiation level, but there is also a large part of gene regulation at the post-transcriptional level.
So was it like 40% of all genes, or was it 40% of the genes? So was there a difference along the genes, or was it just a difference in amount?
For I think there was 37% exactly of the gene that saw a significant delay between the time they were transcribed and the time they were translated. And among this one, there was some that had a small amount of delay, a few hours, and some that were actually translated 6 or 12 hours later. So
what I'm getting is what kind of genes were those, or what kind of proteins are those?
So those genes were genes that are involved in parasite invasion, and also genes that were involved in immune invasion. So a lot of these transcripts are important that are parasite specific first, and that are also very important for the parasite to escape the host immune system. So there was a specific enrichment of this gene family that are parasite specific, that need to be regulated tightly.
You then analyzed again nucleosome positioning in the asexual and sexual stages of the parasite's erythrocytic cycle. Complicated word. And using CHIP, CHIP has not at least been mentioned in this episode. And of course, if you are talking about epigenetics, CHIP must be mentioned. So why is nucleosome positioning so interesting exactly here in this context? And what did you find then using CHIP?
So, nucleosome positioning give you an idea of the enrichment of nucleosome and strong positioning of the nucleosome, but you don't really know what type of modification this nucleosome and histone have. So ChIP-seq look a bit more carefully at the different post-translational modification that the histone have. So you can look at histone H3K9 methylation, that is a mark for heterochromatin, so close chromatin structure, or histone H3K9 acetylation, which is a mark for achromatin, so for chromatin that's more open and more accessible to the general transcriptional machinery. So we now always combine chromatin structure with IC and ChIP-seq with this trying to identify the modification of this histone across the full genome. So we systematically do ChIP-seq experiments and chromatin conformation capture IC to get a global view of what's going on.
Yeah, exactly. So again, not focusing like on one method, but taking a true multiomics approach then, right?
Yeah, we always combine a lot of different type of multiomic approach, from RNA-seq, ChIP-seq, IC. We also do poly-seq, because if you only focus in...
What does this mean? Poly-seq?
Poly-seq, yeah. Just looking at the difference between steady-state RNA level versus the RNA that's translated. Mm-hmm. So we just talked about that a few minutes ago. So I think that combining all of this methodology together really give you an idea of what's regulated at the initiation level, of what's regulated at the post-transcriptional level. But you can also figure out if a specific transcription factor, for example, or a specific reader, or control specifically some gene compared to the downstream effect that you can see often, because there is different cell cycle progression effect that can also affect gene regulation. So it's always better to combine a lot of this technology and analyze them together to figure out exactly what's responsible for what.
Yeah, exactly. What is cause or what is consequence, and also what you need to do then is like look at different time points, right? You also did that, right?
Yeah. we always looked at different time points and always incorporate different omics approach to really understand what's going on. And we now combine ChIP-seq against transcription factor, this AP2 transcription factor family that the plasmodium genome has, together with histone positioning, histone modification, and again, chromatin conformation capture, all together in different conditions.
Yeah, well,
we can knock down some of our important components of chromatin structure and see what this knockdown do on epigenetic feature or chromatin structure. So, but again, always a lot of different omic approach.
Was there something that you can share, like of general principles? Are there some like stages of like chromatin opening and then gene expression that you see for specific gene families or something like that?
So yeah, it's often more or less the same thing. The human malaria parasite Plasmodium falciparum has approximately a 48 cycle between the time the parasite infect a red blood cell and the time the parasite leave the red blood cell to infect another one. And once the parasite gets into the cell, it take the parasite a few hours to actually settle down and try to figure out what's going on. So this is called the ring stage. And after 12, 18 hours, this ring stage starts to actually increase in size. And that's at that time that you need a massive transcriptional regulation to happen for the parasite to grow, of course, and start translation, but also to start dividing into and to start multiplying his own cells. So when you usually have one merozoite that's going to take 48 hours to differentiate into ring, trough, and schizones, and generate 16 to 32 new parasites. So you need a large amount of transcriptional regulation happening when these parasites grow at the trophozoite stage and when the DNA is dividing too. So you have an explosion of transcriptional regulation happening at the trophozoite stage. That's actually the time where you really see a huge change in epigenetic and chromatin structure. And eventually, as soon as the parasite progress into more advanced schizogony and get ready to invade new red blood cell, they are called merozoite, and this merozoite will need to invade new red blood cells. So you have a lot of gene involved in invasion process that are parasite specific that need to be specifically transcribed and translated at the time the parasite is ready to invade new red blood cells. So that's why it's a very tight control of gene regulation to make sure the parasite can first set up into the red blood cell, then activate quickly all of his machinery to regulate energy, DNA replication, and parasite division, and then invasion into the new red blood cell. So all of that is tightly controlled by a combination of transcription factor, epigenetic feature, chromatin structure, and also a lot of mRNA binding protein that are going to stabilize some of the transcript to be translated only when needed.
Mm. So what you then did was that you wanted to find conserved and species-specific principles of genome organization. And first you only looked at two, like Plasmodium falciparum and Plasmodium vivax. And then you generated, you performed Hi-C experiments and generated 3D genome models for 5 Plasmodium species. So I didn't know that there were 5 species, but
there's more than that. There's more than that because there's
obviously,
a lot of different Plasmodium species for every single organism, and not every single organism, but yeah, there is a lot more Plasmodium.
So what did
species.
those experiments reveal?
So, and we're still trying to generate far more chromatin structure feature in other apicomplex and parasite. We've been doing that type of work on 5 plasmodium species, but toxoplasma, also Babesia. We're doing far more Babesia, and we have also recently done a chromatin structure conformation analysis for Cryptosporidium, which is another apicomplex and parasite that can infect human. And the idea was to try to figure out how this genome organization is characteristic of this apicomplex and parasite compared to human. So human 3D structure and genome organization is very, very complex. Because we have a much more complex genome, we have a lot more transcription factor, a lot more of protein involved in epigenetic regulation, I will say. But I wanted to figure out what can we learn doing this chromatin conformation structure analysis, and can we identify a specific feature in Plasmodium versus human? And we did actually identified very particular feature that regulate gene involved in immune invasion. So that's a particularity for the human malaria parasite to survive inside the human host, can survive weeks and months because he has some specific gene family that are specifically designed to evade the host immune system. Each time the host immune system generates new antibodies to actually attack the infected red blood cell and kill the infected red blood cell, the parasite switch the gene to another one. They are called VAR gene, antigenic variation, and then express another of these protein at the surface of the infected red blood cell. So this VAR gene and this gene involved in antigenic variation are very tightly controlled at the epigenetic and chromatin structure level. And when we did this 3D structure of different Plasmodium species or even Babesia species, we actually identified that every single gene involved in antigenic variation are tightly controlled at the chromatin structure level into this heterochromatin cluster that really repress most of these genes to only express one. So one will move away from this heterochromatin repressive center to be actively transcribed when needed, and when the host immune system recognize this antibody, then the parasite will switch. So there is a complex network of this gene involved in antigenic variation that are really tightly controlled using a combination of transcription factor, repressor, chromatin remodeling enzyme, chromatin modifying enzyme to control specifically this gene family. And by doing this IC, we actually identified that these gene involved in antigenic variation are into one area of the nucleus, connecting with each other, interacting with each other, and they may most likely communicate to actually figure out which one will be activated and which one will be silenced.
Yeah. So just a few days ago, I talked to Bas van Steensel and his famous lamina-associated domains, which are mostly heterochromatic. Are those heterochromatic regions in Plasmodium also like localized at the nuclear lamina, or where do they localize?
Localized at the nuclear membrane, but the apicomplex and parasite and Plasmodium species don't have what we call the classical lamina protein. So my lab is actively looking for this type of protein. Do they have a lamina-like? That's the work that I'm trying to do for the past few years now, trying to identify this lamina-like, but it has, so far we have potential candidates, but we still need to validate those. But yes, these heterochromatin are strongly associated with the membrane, but we don't know to which type of protein they are interacting with. Okay. So we're looking.
So another factor in chromatin are long non-coding RNAs. And we also looked at the regulatory roles of long non-coding RNAs in Plasmodium. Can you talk about this and what you found?
So that's where my lab is really going now. We are identified significant change in gene regulation, identified also an important role of epigenetic and chromatin structure. So now we are really looking for parasite-specific components that regulate this feature. And among those, we're looking for lamina-like protein, and we're also looking for long non-coding RNA, because we know these long non-coding RNA are essential in higher eukaryote to recruit transcription factor and to also recruit chromatin remodeling enzyme. Or modifying enzymes. So for everything that I'm doing, we're again trying to do a genome-wide approach to identify which long non-coding RNA are present, which one are enriched in the nucleus versus cytoplasmic fraction, and how they interact specifically with the DNA. So we developed, we adapted a methodology called ChirpSeq to actually pull down this RNA, DNA, so non-coding DNA interaction, and see if there was any specific correlation between epigenetic feature and change in gene expression between the presence of this long non-coding RNA or absence of this long non-coding RNA against across different time points of the parasite life cycle. And we have identified a few candidates that have been validated, but this is a very complex type of work, because so far my lab and others have identified over 2,000 specific long non-coding RNA, and we now really need to figure out what are these long non-coding RNA doing, and are they working in collaboration with transcription factor or chromatin remodeling enzyme to actually control gene regulation. That will also explain a lack of classical transcription factor in malaria parasite or other apicomplexan parasite.
So would you expect...
So again, global view of gene regulation.
So would you expect a more role in transcription or more in structural features of the genome?
I expect both. I think they have a role in both. We're looking, so we know that long non-coding RNA are essential to actually regulate this VAR gene. There's some that have been identified in by many lab in the fields, and we are looking for additional long non-coding RNA that will be responsible together with protein to regulate this heterochromatin structure and to keep them where they need to be. Close to the nuclear membrane of the parasite.
And just earlier this year, you also looked at what kind of proteins are bound to long non-coding RNAs or RNAs in general, proteome-wise. So you did, again, probably mass spec. So what did you find there?
So we have identified a large number of protein, and the idea of doing this experiment was to find this lamina or lamina-like protein.
Yeah, that's what I thought.
And we worked on a few protein, including a PF protein called MORC that have been identified in plants, very important to regulate heterochromatin structure. And we have a paper that should be published soon. I mean, it's already on the website in eLife that characterize one of these protein called MORC, PF MORC, that's important for heterochromatin regulation, seems to interact with a lot of these VAR gene that need to be repressed, or seems to interact with a lot of promoter of the gene involved in invasion to control them and to control their activation a bit later during the cell cycle. So we're really trying a lot of different type of molecular approach to identify this protein regulating heterochromatin structure and VAR gene regulation, and eventually interacting with of course long non-coding RNA. So MORC was one of the first one that we validate as important, and we're looking for, again, the goal of my lab is to identify new drug targets. So we're looking a bit more for parasite-specific components, because there is a lot of histone acetyltransferase, histone methyltransferase, or histone methyl, or desacetylase or demethylase, but they are pretty conserved when you looked at their active binding site, conserved with humans, and while they work well, inhibitor against this enzyme will work well at killing the parasite, there is a possibility of toxicity in human cells. So my lab is really trying to identify these proteins that are more parasite specific, to basically target an enzyme that or protein that can be found in the parasite and not in the human host.
So I guess You already answered my next question, but I want to ask it nevertheless. Maybe there's something that you didn't mention. So imagine you're due to submitting a grant proposal tomorrow. What would you have written into that?
Always submitting grants. So yeah, it's not too far to say that I'm submitting a grant tomorrow. But the work in my lab is really trying to understand how the parasite control its life cycle and how we can stop this life cycle progression. And the idea is not to look at enzymes that are conserved across eukaryotic species, including human, but really to find specific regulator of gene regulation. And if we find this lamina lac protein that will be very different in plasmodium compared to human, or if we identify long non-coding RNA, and the long non-coding RNA are actually very specific. So now we could eventually imagine RNA drug, RNA that will target this long non-coding RNA. The plasmodium genome is very heterogeneous, so kind of far away from the human genome. So identifying inhibitor or molecule that will target and degrade non-coding RNA will be an exciting new way of killing the parasites, and specific. Eventually, we still have to be careful about toxicity issue. This has to be a work in progress, but yeah, looking for components that can be targeted and are specific to the parasite gene regulation processes.
So for the last 36 minutes, we have been on a journey through your scientific career. Did we miss something important, or would you like to add something?
No, I think it's amazing. We went through a lot of the work that I generated over the past 20 plus years. No, I mean, that's been an exciting journey. We really learned a lot more about gene regulation in all organisms, but I focus mostly on the human malaria parasite. And every time we develop a methodology, we just realize how much more we need to learn about the parasite. And I think the biggest challenge we have to face now is trying to understand what these long non-coding RNA are doing to the genome and how they regulate gene expression. They most likely work in combination. It's not only one long non-coding RNA, so it's going to be very, very difficult to figure out what these molecules are doing. So it's the biggest challenge that I am going to face over the next few years, but I'm ready for the challenge. And the goal is to identify new drugs. So my lab is also working on the identification of natural product that could inhibit parasite-specific processes. We're looking at inhibitor of chromatin structure eventually, but also inhibitor of specific organelle, such as the apicoplast. The apicoplast is an organelle that it's parasite specific. It's a bit similar to the chloroplast in plant, but not exactly. And this is also something that the parasite has and the human host don't. So looking for drug, and we have so this is also something that I'm actively looking. We're screening natural product. We're focusing on natural product, thinking that that nature has done the work to identify compounds that are specific to infectious agents. So this is also something my lab is doing. Mm-hmm. So we're not only looking at understanding gene regulation at a basic level. We're also doing a bit more applied science to identify drug that can kill the parasite in a very specific manner.
So my last question, and this might be very interesting because you're looking at the whole thing from maybe a different angle than most of our guests do. So what is your personal definition of epigenetics?
Personal definition of epigenetic. It's it just control life. I mean, I think this is the most important molecular process in any cell, regulating life. So it's not DNA sequence, but DNA sequence without epigenetic doesn't do much. So you really have to understand epigenetic if you want to understand life.
Yeah. Thank you, Karine, for your time and for being on this show.
Thank you so much for your time too, and thank you for the invitation.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.